I'm Camilla Bellone, Assistant Professor at the DNF since 2014, and this is my research group. Our research focuses on the role of a reward system in neurodevelopmental disorder characterized by social deficit. In mice, we combine behavioral and electrophysiological experiments to characterize forms of synaptic plasticity associated with social experiences.
Hi, I'm Paola Bezzi, Senior Researcher at DNF since 2008. I have been studying the role of astrocytes in the brain communication system for several years. In my group, we are combining a variety of complementary techniques such as biochemical analysis, in situ immunolabeling, and behavioral approaches to discover aspects of astrocyte-neural interactions that are critical in the formation of neuronal circuits in the developing brain. And thus, we would like to reveal whether astrocytes are involved in the genesis of neurodevelopmental disorders.
I'm Jean-Yves Chaton. The research of my group focuses on the functional interplay between neurons and glial cells. We want to better decipher how energy metabolism, neurotransmitter regulation, and ion homeostasis influence neuronal activity. To gain access to spatial and temporal aspects of these dynamic interactions, we develop fluorescence imaging approaches and combine them with electrophysiology. These functional neuron-glia interactions are central to several brain pathologies such as epilepsy. Our studies find applications for therapeutic interventions for such diseases.
I'm Isabelle Descoster. I'm one of the PI of the Department of Fundamental Neuroscience, and I'm also a clinician at the Pain Center of the CHUV. The main goal of my group is to better understand the mechanism of chronic pain. We are studying the deregulation of activity at the periphery that will lead to maladaptive plasticity and persistence of pain symptoms. My role is not only in research, I'm also taking care of chronic pain patients in the hospital and teaching at the medical school and the biology school of the faculty.
My name is Lawrence Hurk and I am a neurologist specialized in stroke. Our lab is part of the Department of Clinical Neurosciences and the Stroke Center of the CHUV. We are studying stroke using an experimental approach. Our aims are to better understand mechanisms of cell death after stroke, to identify diagnostic biomarkers, and to develop new strategies to protect the brain and improve the behavioral outcome after stroke. At present, we are focusing on the thrombin PAR1 junk pathway and the neuroprotective role of lactate. And our main goal is to translate our findings from the bench to the bedside.
My name is Jean-Pierre Hornon. I am professor at the DNF, and this is my team. We are investigating the long-term consequences of the inactivation during development of the 5-HT1A receptor, a serotonin receptor implicated in mood disorders in humans as well as in animal models. Using morphological and physiological methods, we have observed alterations in the connectivity and synaptic plasticity of the hippocampus, which are more prominent in male than in female knockout mice, a difference that can also be seen at the behavioral level. We currently analyze the associated cellular and molecular mechanisms and validate that the 5-HT1A receptor expressed in the forebrain specifically contributes to this phenotype.
Sleep is what we are mostly concerned with in our laboratory. Sleep is a very active brain state. The sleeping brain does things that tell us a lot about our physiology, our equilibrium, our capacity to learn and remember, and about the origins of many diseases. We also look at sleep from the point of view of a plastic system. How do electrical sleep rhythms contribute to the plasticity of the brain? And what are some of the mechanisms involved? Finally, we look at sleep from the point of view of large-scale circuit connectivity. How do sleep rhythm-generating circuits interact with other brain areas, such as those involved in learning and emotion? and what kinds of activities do they generate together?
Hi, I'm Anita Tretman, pediatrician specialized in neonatology.
Hi, I am Julian Puel. I am a senior researcher here at the DNF. I am interested in understanding neuronal death mechanisms occurring after neonatal cerebral hypoxia ischemia. The overall aim of our research is to find new targets for neuroprotection in neonatal cerebral ischemia and then to propose in the future new therapeutic strategies at the clinical level.
Our group associates a basic researcher and a clinical neonatologist with the objective to bring together basic and clinical insights for the development of neuroprotection in babies.
Hi, my name is Romano Regazzi. I am professor at the University of Lausanne since 1997. My group is investigating the contribution of non-coding RNAs in the control of pancreatic beta cell functions and in the development of diabetes. For this purpose, we use molecular and cell biology techniques to identify the non-coding RNAs that are differentially expressed under physiological or pathological conditions and to study their role in insulin secretion, in beta cell proliferation and in beta cell survival.
Hello, my name is Nicolas Tony and I lead the Laboratory of Adult Neurogenesis in the Department of Fundamental Neuroscience. So this is my group, and together we're trying to understand why the adult brain retains stem cells. Neural stem cells are found in the hippocampus that plays a crucial role in learning and memory, and is impaired in Alzheimer's disease. They are surrounded by many brain cell types called the neurogenic niche that we are trying to manipulate in order to understand how adult neural stem cells are regulated, with the hope that one day this knowledge can be used for brain repair or the treatment of amnesia.
The Volterra group is interested in a recently discovered form of communication in the brain between neurons and astrocytes, and in its contribution to the integrative properties of brain function and the pathogenesis of brain disorders. To this end, we perform several complementary levels of analysis using correlative light electron microscopy, 3D two-photon imaging, genetically encoded calcium indicators, optogenetics and electrophysiology, miniaturized microscopes in freely moving behaving mice, and astrocyte-specific mouse genetics. We expect to demonstrate how astrocytes participate in cognition and how alteration of their communication contributes to the progression of neuropsychiatric disorders, thereby identifying completely new therapeutic avenues.
We use the whisker-to-barrel pathway in a mouse to study adult plasticity. Whiskers are sensory organs implanted in the skin and are innervated by the trigeminal nerve that reacts to the brainstem, where neurons relay information to the thalamus, from where there is a projection to layer 4 in the cerebral cortex, where there are barrels. Barrels are formed by neurons that have dendrites with spines and dendrites that have no spines. We have shown in the past that increased sensory stimulation inserts carbohydrate synapses on spines.
Here we have an example of combined in situ hybridization and immunolabeling of PSD95. There is no change of this microRNA after simulation, but they're redistributed and gets closer to postsynaptic sites.